Motif. Thank you for joining us for the second PIXUL user meeting. This webinar will focus on proteomic application. Today, we have the amazing opportunity to listen to Dr. Alexander Schmidt, head of the Proteomics Core Facility at the Biocentrum in Basel in Switzerland, and Dr. Simon Devos, Proteomics Core Manager at the VIB in Ghent, Belgium. Alexander, the floor is yours.
Thanks a lot. And Sophia will just, okay, exactly. Share. Okay, so I hope you can see it. Okay, yeah, it's a pleasure to tell us something or tell you something about the PIXUL, so how we're using it. I just checked, actually, I thought we only have it like for 2 years, but it's already almost 4 years that we have the PIXUL running in our lab quite successfully. And I want to tell you something about how we're using it and yeah, something about the experience and why we actually decided to go and use the PIXUL. So yeah, first introduction to our facility, exactly why we purchased it, and then I mean some applications that we did with the PIXUL or using the PIXUL, particularly for large-scale experiments. We also tried a few tests in the beginning, like different lysis buffers. I mean, as PIXUL is mostly used for sample preparation, I also want to show you something what we tested, and that might be useful because it's all about mostly about sample preparation. We also have some challenging samples from our users, mostly bacteria, and therefore the PIXUL turned out to be really useful. So I want to share some data with that. And then we also lately went more into the area of low input samples. And there I think also the PIXUL is useful, although I think there might be something that can be improved. Okay. Introduction. So we are the Proteomics Core Facility at the Biocentrum. This is part of the department at the University of Basel. So we're located on this side of the Rhine. Of course, you know, I mean, Novartis, Roche, but also the Friedrich Michel Institute. They all have proteomics labs, but they only have that for their own research. So we are the only facility that actually works for the whole university. And we also have a lot of customers from startups, so smaller companies that don't do proteomics. Yeah, so last year we had around 10,000 samples processed. So I would say this is rather a lot, which is getting more or easier actually with the new machines, but also because of the PIXUL, of course. Yeah, and there's a nice campus. So we're located here, and there's like a children's hospital. We have the university hospital. We also work a lot with the Department for Biomedicine. So we get a lot of clinical samples, like really larger samples. So basically we get all kinds of different samples that we need to process. And we have a new building since 2021, and we are lucky to be actually at the 13th floor. It's here. So we have a very nice view. So I can not complain. Yeah, so as I said, we have very different kinds of samples we have to process. It's a basic research institute, the Biocentrum, but as we work for many other groups, we also have a lot of clinical samples and others. So yeah, we have tissue, cell culture, microorganisms, body fluids, and also single proteins. And just to show you how we do that is the first step and the most crucial step actually for having a reusable proteomics analysis, the protein extraction. So you extract proteins, you roughly end up, if you let's say work with human tissues or cells, you end up with around 10,000 proteins. Next step you do is you digest the proteins into peptides. This is that facilitates actually the analysis, but of course it increases the complexity of the sample. So instead of 10,000 proteins, you end up with more than 100,000 or even 1 million different small molecules you have to analyze. They're usually in the range of 7 to 25 amino acids. And then comes the mass spec analysis. So I said this very complex sample. First thing we do, and this thing that every proteomics lab is doing that, you decrease the complexity by separation. You use liquid chromatography for that. Then you ionize these peptides, means that they go into the gas phase, so then liquid here in the gas phase, and they fly into this mass spectrometer, to the detector, and get also charged. We usually have orbit traps that measure the mass of the peptides by the speed, how they cycle around the spindle. But there's also new instruments that have TOF, so they measure the time, how long it travels. So they're also very interesting and new. And then you have a data system that actually acquires the data, stores it, but also analyzes it. And you can actually, these peptide sequences can then be assigned back to the original protein, and the intensity of the signals that every peptide derives can be used to compare the amount of a protein between samples. So this one has, I mean, in the last years, like every step was really optimized, the mass specs, there's new hardware out now, which is really fast and sensitive. I will come this to the end. Improved very fast. Data systems have been improved. So everything is now to analyze the data is actually much more improved. Also separation techniques, there are new LC techniques that can really separate the peptides much better. So that allows actually within half an hour now to derive or to identify at least 10,000 proteins. So this is all fine, but you also have to make these samples now. We can very fastly analyze them. And I think the sample preparation is something which is harder to automate. And it's so far, I think, the least coming now, but I think it's something that is so far it's not so easy to automate, and I think not so much automated. So that's something we really focused on the last years. That's also where the PIXUL comes in. So just the workflow, we are very fond of this sonication to lyse the cells, because if you use probes or so where you have to go in, we often have bacteria samples, and then also for safety reasons, not the best option to do. You can work with rather small volumes. So having like ultrasonication to lyse cells in a rather small volume is a very good option. And we started out with the Bioraptor Pico beginning, so they can handle up to 12 samples at a time in 0.5 milliliter Eppendorf tubes. We measure then the protein concentration with we started out with a fluorescence scanner, because before we were using BCA assays or the colorimetric assays, and there you need reagents. It's also a bit cost intense. And there was a nice method published. I can really recommend that from Wisniewski, from the MAN lab. And they use tryptophan fluorescence as a control, but also to measure the tryptophan content in proteins to measure the protein concentration of the samples. So this is very robust, and it basically doesn't require any reagents, so it's very fast and very cheap. And we also use this to measure the peptide concentration with UV or with a colorimetric assay. Then we have a bunch of liquid handling platforms, the Ashland Bravo. We have a Tecan Fluent for liquid handling, and we have a A200. These are all used to automatically process samples. We use the Proteomics IST kits. We use SP3 S-trap methods, and also IMEC for phospho enrichment, mostly with the Bravo. And then they all go into the MS systems. We have mostly Vanquish Neos, Orbitraps 480s, Eclipse Lumos, and since a year now, also timsTOF Ultra. And since we have run a lot of samples, we also have this EvoSep1 LC system, which can process, I mean, like, it's really, really high throughput LC machine to be much faster. So that's all fine, and this works great. And I mean, if you want to analyze a lot of samples, you have to go for multi-titer plate format. And if you look at that, all machines are compatible with that, except this Pico Bioraptor, of course. So of course, obviously, this is the bottleneck in this sample preparation pipeline, and that's why we were interested in purchasing a PIXUL. And we also started out and tested a few other vendors, at least actually mostly one, that can actually do it, because it's not so easy to have the same, let's say, force. So you need a bit special sonication device that actually puts the same amount of force to every well in 96 well bed. So in a simple ultrasonic bath, this will not work, because every well will experience a different type of amount of energy. So we tested that. We took yeast cells, also some other tissue samples, but the results were very consistent. So I just show you the results for the yeast cells. We lysed them for 20 minutes. We used, as I said, different devices, also different buffers, and then we analyzed them by mass spectrometry. And these were the results that we got. So if you have a look at the buffer 1 system, so extraction method 1, I mean, seems to be a bit higher than the other ones. And we tried the PIXUL, the Bioraptor, and another company. I have to say, the aim was not to be now much better than others. I mean, we were very happy with the Bioraptor. The only aim that we had was to increase the throughput and to be more consistent. And what you can see is actually that the PIXUL in the buffer 1 at least is extracting or identifying a few more proteins. But what is more important is that the amounts were very consistent. And the Bioraptor had a bit more variation, and the other machine also was actually very consistent, so it was also fine. We used another buffer system, again, the same picture. So you have PIXUL performing very well with like very consistent identifications, Bioraptor a bit more variable. This buffer didn't work for the other machine, but they had their own buffer, and with this it worked really great. Yeah. So I have to say the other machine that was also in consideration was also fine. It works totally good. But the difference was that it was much more expensive, and also particularly the running costs per plate were much higher. And that's something that decided us to go for the PIXUL, because the plates are very cheap. We can hand them over to our users, and they can fill in the samples, the lysates, and then, or the cell pellets or samples. We add the lysis buffer, and then we start here with the PIXUL. I just want to show, because one thing that is maybe a bit different to other systems is this liquid, this coupling fluid that you have to use. It's a bit sticky. In our case, what we do is actually after applying the sonication, we quickly put in the water bath for, I mean, like 10 seconds or so, and then we spin it down, and then it's usually gone. Yeah, so this is not really a big problem. So that was fine. So it looks like everything's working fine. So we use that. And we use it in combination with the S-trap. I just briefly want to mention what this is. So for those who don't know it, so the S-trap is like spinning, not this, but a spinning capsule basically, where you can trap proteins inside this resin. It's size-based, and the nice thing about this, it's very fast, and it's compatible with almost all lysis buffers. So you can also use SDS, which for most other workflows is not compatible, but SDS is working there. So you add the samples, the proteins are bound to the spin columns and trapped because of their size. And then you can wash everything that is smaller or small molecules, you can wash away. So you get very clean samples. You add trypsin and the digestion buffer. Trypsin itself is a protein. So it's trapped as well. So that's why the concentration of the trypsin is very high. And the digestion is very fast. So in 1 hour, you're good to go. Peptides are small and elute from the system again. So we use that particularly for pilot studies, because there is a 96-well format available, but this is only for high amount so far. The low amount version, the micro format, is not available. So as a plate, so we use it still, but just for, let's say, up to 15 samples, particularly for pilot studies, to set up the whole workflow, because it provides really, really clean samples. And it's fast and easy to do. And the workflow we use more for a higher number or larger projects is the so-called SP3 workflow. So single pot reactions. Here you have the proteins also in the solution. You add magnetic beads, and then you bind these proteins to these magnetic beads. You can actually then use a magnet to trap all the beads and the proteins. You can then wash all the contaminants and small molecules away. You add an enzyme and a digestion buffer, and then you can elute these peptides and then measure them. We use this Tecan, the fluent from Tecan. Again, it's compatible with SDS. It's a very cheap workflow because these magnetic beads cost almost nothing, which is really nice. It's also very nice to automate, but it's actually really good to have a robot for that, because it needs a lot of pipetting steps, and that's better done with a robot. But it also requires an overnight digestion, so it's not so fast, but it can handle also quite low amounts. And we use it basically for all projects that have more than 15 samples. And the whole procedure now on the Fluent, because it can pipe it very quickly, is like 30 minutes to prepare a full 96-well plate. And I just want to show you, I mean, for us, it's really perfect because the PIXUL can, so we use usually 80 microliters of lysis buffer in the PIXUL, and we set up the workflow in a way that it's compatible directly with 80 microliters. So we can directly use the PIXUL plate, put it into this Fluent, and then we can actually now have also integrated this fluorescence scanner automatically. So it takes out a certain amount, makes the protein measurement. This data goes in, it calculates how much volume is needed to take out to derive. Usually we take 10 micrograms. It fills it up to 80 microliters, and then it does the whole SP3 protocol. So this is something that we're trying to get to a fully automation, although it's hard, but we're trying to get there. We also we can also do peptide concentration measurements and then make a dilution, load this also on Evo tips. There's also Tikan and EvoSep are working together, and there's a new protocol on this, how to automatically load Evo tips, something also quite useful. And we're also working on an S-trap analysis. Just on a video here. So this Tikan fluid is very fast, and the trick, or what is really nice about this, is it also has a liquid detection, because SDS, for example, tends to form bubbles, and then it can actually detect the liquid, and then just with the liquid increase the height so that there's not much turbulences and bubble formation. So we can even pipette 5% SDS without any bubbles. That's very nice. Okay, so some applications. So one thing we tested when we got the PIXUL was different extraction buffers. There was also with the discussion, I mean, what we can use, and now that we have SP3 and also S-traps, we can actually use almost every extraction buffer. So the idea was that we try a few and see what works best on a global scale. So it turned out that for higher eukaryotic cells, the lysis was not so hard, and I mean, to lyse them, actually almost every lysis buffer worked quite well. But for bacteria, we saw a bit more differences, and I just want to show you that. So we took, in this case, E. coli cells, and we tried 7 different buffer systems. We also tried different temperatures. SDS was used with other temperatures. Otherwise, urea, we only used 37 degrees, but all others were also boiled to 95 degrees. We did this in triplicates, and then we did mass spec analysis of it. And you can see that, I mean, based on the protein amount, it actually looks quite good. So we get rather equal number of proteins. We used Epigal and, for example, Triton, which are non-ionic detergents. The oxycolate is this PO when we had Rapid Chest, which is also a detergent that is used, and it's actually working quite well, compatible with mass spec, because in acid it cleaves off, and then you can easily get rid of it. Reaper buffer is a mix of deoxycholate mostly, but also has some SDS at different temperatures, and urea is something which is, I think, no longer used much, but still is very good for solubilization of certain proteins. And if you look at that, you can see that these buffers, they all cluster together, means that every buffer extracts a certain set of proteins. And I just want to highlight this. I mean, these non-ionic detergents are not so strong when it comes to so these are only membrane proteins. There we saw the highest differences. So these are proteins that are mostly involved in transmembrane transport or symport activity. There's not a cluster for deoxycolate, which is not so good for ion transport, amino acid transport, so also membrane proteins. But for example, here is a cluster which is where deoxycolate and also Reaper buffer is actually really good. And these are outer membrane proteins, so these buffers are really good. But for example, this part of the proteome is more inner membrane proteins, ATP hydrolysis activities, also ATP synthase, which is really specifically well solubilized only with SDS and only when it's boiled. You can see it here. If you don't boil it, you get it here, but if you boil it, you get a much higher extraction efficiency. So yeah, I mean, this we also see actually at the single protein level. I don't want to go too much in detail, but you have some proteins for some reason that are very good solubilizable with a certain buffer, and others proteins are better with another buffer. So it depends a bit what you're looking for. I also want to mention that I mean, of course, you see these differences, but it doesn't mean that if you use another buffer that you will not be able to identify this protein or quantify it in a way that you compare it. Yeah? So if you would extract always a similar amount of this protein, so if there are changes in this protein, you will still be able to see them. But if you do absolute quantification, so if you really want to know how much of this protein is in your sample, then of course an efficient extraction is really key. Otherwise, the amounts are too low. Okay, yes. So we decided actually to use SDS buffer at 95 degrees because this seems to be having the least biases, and it's ready available, it's cheap, it's also very much used. So we stick to that. And we used it for, let's say, a bit more challenging samples that we had. And one is this Mycobacterides abscessus. It's a project from the medical department. It's a human pathogen. It's rapidly growing. It's also multi-drug resistant. It causes infections of all organs, mostly lung infections. And this is really nasty because it's, even if you boil it for 20 minutes, there are still some bugs that survive that. And it's super, super hard to lyse. Yeah, so it's a good challenge for the PIXUL Sonicator. And we started out using the Bioraptor. And if you compare it, so this is the Bioraptor for a certain amount of cells. And we actually did, I think this was, was higher amounts and these were lower amounts. So the concentrations you get are usually a bit higher with the PIXUL as what we got with the Bioraptor. And as you can also nicely see, is actually the Bioraptor again, it seems to be more consistent and more robust and comparable compared to the Bioraptor. Yeah, because here you have really large differences. And you see the same also if you run this on the mass spectrometer and you look at the correlation of the protein profiles, you can see clearly that the PIXUL replicates show much higher correlation than if you use the Bioraptor. Yeah, so this seems to be more precise and more consistent for across the different samples. And this is an application we did then. We run 360 clinical isolates and define the proteome. This is for a proteome-wide association study. So we want to link, or they want to link actually the phenotypes and genes for functional variations. And we get really very consistent identifications. I also can show, I mean, I didn't show that, but I think it's true that the amount of proteins we extract was also very comparable across all these samples. Yeah. We can also see that we get some clusters already for some certain samples, so that worked really amazingly well, but the data analysis is challenging and we're still working on this. Another project which is increasingly coming is this so-called deep visual proteomics, although I have to say we don't run a whole tissue, but I mean, what people are interested in this case, Julia, so they are interested in certain parts of tissue. So there's a lot of light microscopy done here at the Biocentrum, and people see some areas of interest that they want to know what this is or how they differ from other parts. So what we did then, we can just take a tissue, you can identify a certain area of interest, and then you use a so-called laser microdissection to actually cut out certain pieces. Like here, we use the size palm, it's a rather old instrument, and you can cut out certain pieces of different areas you're interested in. And this is a rather small amount, although you can cut multiple times and increase the amount. And then you can subject that to proteomics analysis. And in this case, she was interested in this myotenonous junctions. And they're in the group that works on a muscle dystrophy mice. So they have a certain phenotype. And the idea was to see what is the difference in this MTJs between the control and the disease mice. And what you can see is that we get a lot of proteins, although I have to say in muscle, it's difficult to get a high number of proteins because there's the same as in plasma. You have a few very abundant and very large proteins. For example, this titin protein, also the myosin proteins, but the titin protein in particular makes up like 50% of the whole mass. It's the largest gene actually in the human genome. So you mostly analyze peptides of titin and not of the actually proteins of interest. So 5,500 proteins is quite good. And what is more interesting is actually that we get a really distinct the clustering of the samples. So of course you get a difference between muscle and MTJs, but you get also a nice separation between the control and the dystrophy for both cases. So there was a lot of proteins that changed, and Julia is now following up on this. I want to finish with something which is really coming more and more in the proteomics field. If you look a bit at the history, so there's a lot of inventions. And lately, now, as said already, there's a few improvements that really boosted the whole field. So in separation, in DIA method, which is really focusing or analyzing almost the complete mass range. And also this latest development of these new instruments, as at this time of life instruments, that all speed up the whole analysis in speed. So you can make up now, and what you could do before took like a few days, you can probably now do in a half an hour time. And the other thing is also, it's not only much faster, but it's also much more sensitive, these instruments. And what we have now for a year is this timsTOF Ultra instrument. This is how it looks like, with like beautiful eyes. And the reason why it is so sensitive is it's basically compared to the orbiter we usually have, it needs a lower number of molecules to give a signal. And also, what is the advantage of this TOF instruments is that no matter even if you measure very fast, the peptides or ions that actually have to be measured travel the whole way. So the resolution is not altered even if you measure very fast. In contrast to the orbiter, when you measure very fast, the quality goes down. So you basically have a high quality scans, and you have also you don't need so many ions, so you're also much more sensitive. And that allows basically now totally new workflows. And I just want to mention that. So if we don't have any challenging samples, meaning, for example, in this case, brain cells that we want to compare, the workflow is really straightforward. And you also try to minimize the amount of steps you do to pipette, because every pipetting step might lead to losses. And you want to work in a very small volume. So in this case, we take FAC cells. So Violetta, who is driving this project, is actually interested in a rare species of brain cells. And she did FAC sorting and directly sorted them into 8 microliters of a lysis buffer. And this contains only DDM and some TF buffer for the pH. And only 100 cells were sorted. And then you just put this with a bit of trypsin for 3 hours at 37 degrees. And there's no other step that you do. And you directly run that in the mass spec. And that gives really amazingly results. So we got more than 5,000 proteins. And as I said, for their different conditions and cell types, we get really nice data to work for. So the question is, of course, if we can also go down to 1 cell, and people did that. And when we do that also with the same workflow, this is roughly what we get. So we get almost 2,000 proteins. But if you have a few more cells, you get more in the range of like 3,000 to 4,000 proteins. That's something which, yeah, you can then definitely, yeah, have a higher coverage or enough coverage to draw some biological conclusion out of it. But if you have more cells, of course, coverage is actually even improved. Yeah. So I just want to show that because this type of analysis is possible, but it only works for easy to lyse cells. We have other projects which are more and more coming, which are challenging to lyse. So you would need some mechanical force, and you cannot do that without actually, yeah, making too many steps. So I think a Sonicator that would work with much more smaller volumes would be really, really good to have. Yeah, in this case, it's just one experiment we tried was, again, a bacteria, Staphylococcus aureus, which is really also very hard to lyse. So it needs a mechanical force. You cannot just boil the sample. It will not lyse. And we have to work in small volumes, but in this case, we didn't do it. So we had to work with 80 microliters. And as another alternative approach, which does not require, or which works in smaller volumes, and is also particularly designed for this bacteria lysis, is the so-called speed workflow, where you just use TFA concentrated, you incubate for a few minutes, you neutralize, and then you digest the samples. And we compared this workflow to the PIXUL workflow, where we know if we work with higher volumes, it's maybe not ideal, but still, I mean, the PIXUL workflow and the SPF3 cleanup, even for 50,000 Staph aureus cells, which is corresponding to, let's say, I don't know, 20, 30 human cells, you can still get quite some proteins. And you're a bit better than the speed approach, which I think it's difficult to automate. And it's maybe not it works great, actually, because the lysis is good, but the numbers were a bit lower. And yeah, it would be, I much more prefer this workflow. And I think if we can work with smaller volumes like we did before, I mean, the recovery of this workflow could be greatly improved also. So that would be fantastic. Maybe there's something for the future. So to summarize, I think, I mean, now the PIXUL we use it now for almost 4 years. It was very well integrated in our facility. It's very easy to use. We use it, as said, for larger projects. I mean, up to 96 well plates. It's very quickly. I mean, 20 minutes you can lyse a whole plate. It's not so expensive. The plates we can also give them to our users and for them to collect. And if one plate gets lost, it's not a problem. Said some ideas maybe that you have a PIXUL for smaller volumes might be actually really useful for future projects where we have low amounts and difficult to lyse cells. But overall, it's a very nice tool that we have. And I want to thank you for attention. This is the group that did all the work, and I'm happy to answer any questions.
Thank you very much, Alexander. So if you have questions, you can write them in the chat. So we already have some of them. So the first question is, with the yeast lysis test, how much volume of buffer did you use to resuspend your 50 million cells?
So we always use we played around a bit, but we always try to use the minimal, and the minimal volume we use is 80 microliters. I have to say we didn't push it down. It was just a recommendation that 50 microliters is the minimum we could use. That's why we didn't push it. But I've also heard you can maybe use even 50 microliters, but we didn't try that. So we used 80 microliters.
In fact, to reply also to another question regarding the smallest volume you can use with the PIXUL. So we recommend a minimum of 75 microliters to get very good consistency. But you can go down to 50 microliters. Just the consistency is not reaching our criteria. Okay, but you won't damage the device if you use 50 microliters.
Okay.
Another question is, are the protein concentration based on well or 96 well?
If the protein concentration are based on 96 well?
I don't really understand.
So I mostly showed protein identifications, yes, but the protein concentrations are based on this 80 microliter digestion, and we do this in a whole plate, yeah. I don't know if this was the question.
Another question is, can you talk about how you and collaborators get the cells from the culturing dish to the PIXUL 96 well plate? Do you lyse then transfer or vice versa? We have issue lysing in SDS as the genomic DNA makes the lysate hard to transfer. Yeah,
exactly. So yeah, so it depends exactly. So if you use eukaryotic cells, you have a lot of DNA, and if you add SDS, immediately cells will lyse, and then you have this glibber thing. Yes. So if we normally work now with really low numbers of cells, and then this is not so much of an issue. If you have more and you have, let's say, really this, I mean, what we do is actually we then increase the volume, because then we know we have a lot of material. So we increase the volume to a point where we can pipette the samples again. And after the PIXUL treatment, then the DNA is destroyed actually, and then it's nice and fluid. The other thing one can do is also to add a bit of benzoate to the samples. That will also digest actually DNA, and then the samples will also get more liquid and easier to pipette. Yeah, but this is indeed a bit of problem. Sometimes if we have cell pellets, like really high amounts of cell pellets, then this becomes a bit of a problem. Yeah, that's true. But dilution is fine for the mass spec analysis. We don't need much. So then we split it. We also did that. Then we split it in a plate, like in 8 vials, and then we sonicate, and then we recombine it afterwards again, and then it's fine.
And the last question is regarding the volume range you can use with the PIXUL. So the minimum is 75 microliters for good consistency, and you can go up to 180 microliters. It's a 96 well plate, so anyway, the maximum volume is 200. But with the sonication and all the bubbles and bursts inside the samples, we recommend 180 microliters maximum.
Yep.
And last question, post-PIXUL treatment, are you able to do separation of supernatant and cell debris? If yes, what method do you suggest?
So you mean after if you use a PIXUL and then afterwards it's there's still some debris inside? Yes. I mean in the plate of course you can spin down. The plate is not you cannot spin at very high speeds. Yeah, in this case, if it doesn't dissolve too well, yeah, I mean we spin down the plate and then try to take the supernatant. We spin them a bit for a longer time, and if this really doesn't help, then we would also go into like an F tube and spin again to be really clean before we put it into the further preparation. Yeah, that's what we do.
And last question, and then we let Simon speak. Hello, for clients who already prepare their cell pellets, low input material in their own 1.5 tubes. Would you perform the protein extraction DNA treatment in bioreactor or PIXUL, as the latter one would require transferring the samples to a 96 well plate?
Yeah, as I said, I mean, we tried that, and I mean, it's always like the lower amount of steps you do is better, and that's why we directly FACS sort or have the lysis in a plate. If the cells are not so easy to lyse, because we usually use DDM not only because it lyses the cells, but it's also very good for prevent any absorption. Yeah, I mean, if it doesn't lyse, then I think then you have to find a different method. And then I think depends a bit. It might be more useful to go into PIXUL, also maybe use a different detergent and do cleanups. That's something that has to be tested. But for low amounts, usually the best method is to just use a plate and heat the sample so far. But it would be nice to have, as I said, mechanical force. I don't know if it's possible to have some kind of we thought about like maybe a lid that actually goes, because in the end, the volume limit is defined because the volume of the well is so big that all the liquid actually evaporates, or this is the problem. And then this would run dry, and then it would harm the ultrasonication device. And I don't know if you can actually have something like with dents or like something tubes that go inside, reduce the volume actually of these plates. And then you could maybe use even lower volumes. I don't know if this is possible.
So right now we are trying to develop modification in the device to be able to either use a small volume or big volume. But I don't say too much because we are really at the beginning of development. Okay.
Yeah, because this would be really interesting, because I mean, as I said, every step is a potential loss of sample, but the sonication is then everything in the same plate. That would be super interesting. Yeah.
Thank you very much, Alexander, for your presentation and your answers. Simon, I'm happy to let you present.
Yes. All right.
We can see your screen.
Okay. So first of all, thank you very much for having me. And also thank you, Alexander, for your very nice talk. It was also interesting, especially the part on the yeast and the bacteria, because that's sometimes, where we still look at other methods for lysis. Maybe quickly to come back to some questions that were posed while the heat is on. So usually when it comes down to debris, there's very often a centrifugation step involved in the plate itself. So what we often do is centrifuge it at 60,000 G, for example, for 10 to 15 minutes, and then transfer the supernatant of the sample to, for example, the S-trap protocol. And maybe also coming down to the sample of the very low input proteomics. So when we really go to single cells, we don't tend to go to PIXUL because we're stuck with this very low volume. So we often work in 384 plates, and then, well, at least for mammalian cells, the lysis we do with freeze-thaw cycles or yeah, with other types of things, and very often also on a robot like a Cell-in-One system where the whole sample prep is kind of streamlined, let's say, and tailored for these low volume samples. But anyway, again, so thank you. So let me share some insights on the use of the PIXUL Sonicator for prepping samples for proteomics. I'm also glad that Alexander already introduced some of the proteomics kind of basics, so I don't have to bother you with that. I also didn't include it in in my introduction, in my slide, so that was a good guess. Very briefly, introducing the VIB Proteomics Core, so which I'm heading. So I'm Simon, by the way. So VIB is the Flemish Institute for Biotechnology. So we have different centers spread across the Flanders region. So it's a government-funded institute to accelerate research on different topics, for example, like inflammation, cancer, plant systems biology, you name it. And so, well, we're a government-funded institute, but we are affiliated with all universities spread across Flanders as well. But what we also have is a very well-built core facility program. So we have several core facilities, each specialized in its own technology, like metabolomics, nucleomics, microscopy, everything that you see here. And of course, my part is on the proteomics core. So yeah, that's a bit an obvious thing. Our mission is, of course, to provide proteomics, mass spectrometry-based proteomics services to whoever needs it, and to whoever that is willing to pay for the service, of course. Because yeah, you all might know that this technology it doesn't come cheap. And it needs quite a bit of expertise, and a lot of the expertise, of course, is in not just in the infrastructure, but also in the people that are working in the facility. So we kind of, yeah, we can provide all in service, but we also do a lot of training. So, for example, in a couple of weeks we have our yearly training where people actually also learn how to use the PIXUL and following, for example, STRAP. Also, thank you again, Alexander, for introducing STRAP, because it's also a sample prep method that is, for the time being, let's say, our preferred method, but it also kind of depends a bit on the application. So we also have the SP3 indeed, also Amphipulse that we also kind of use sometimes for different applications. So yeah, we also look at, of course, how to implement new technology, develop technology with research facilities next door. And yeah, we also engage in a lot of collaboration, which goes hand in hand, of course, with the technology implementation and so on. So this is the services that we provide. So it's quite broad, of course. Our main services in the facility is still shotgun proteomics, interaction proteomics, and PTM analysis. These are like the most popular services that people ask for. But now more and more clinical and spatial proteomics, of which we have good methods in-house. Single-cell proteomics is not yet in service mode because it's well, challenging for one thing, and also not yet affordable as a service, because well, people don't come with 3 cells of a wild type and 3 cells of a mutant or whatever. So it's hundreds of cells, and it requires a lot of expertise, knowledge, prep, and throughput. And then below, and immunopeptidomics is also something we have invested quite a bit of time in and works very well. And then we have a separate biopharma platform for intact protein measurements and peptide mapping, but that is not, let's say, the focus here. What is the focus is, of course, well, we have sample prep to LCM to data analysis, and the sample prep part is, of course, where the PIXUL comes in. There's really not a lot of samples that we haven't seen before in the in the core facility, especially from human and mouse. I mean, I think about everything from a human or mouse we have seen already and processed it. But also a lot of plants, which also have a challenge on their own. But an important thing to say here is that almost all these samples have seen the PIXUL in the pipeline. Either it's directly for lysing the samples, and DNA shearing, which we also accomplish with that PIXUL machine, or when it's very challenging samples, for example, plants, then we still do an upfront like a lysis step where we either go to bead milling or like plants are often homogenized first by putting them in liquid nitrogen and really using a pestle, but these of course lack a lot of throughputs. So we are looking into ways of improving that also with a role of the PIXUL in there. So what's also mentioned, of course, is the throughput we have there with the 96 well plates. Is very interesting for us, because, well, Alexander mentioned the roughly 10,000 samples per year. We're not far off. I calculated it for last year, it was 8,500 samples. So yeah, if you don't have sufficient throughput, I mean, you really need it in your facility, and the PIXUL really facilitates that. So these are our mass specs that we now have up and running. So the majority are Orbitrap type machines, but we also have one Bruker-Timstorff Ultra. It was actually an SCP before, but we upgraded it recently to an Ultra, and in about a month we will also do a second phase upgrade to the Ultra 2. We're also, so this is our ultra high sensitive mass spec because that one we really use for more low input samples, which I will come back to later for example for spatial proteomics, but also single cell or low cell amounts, let's say. Of course, the Astral is also very popular. We're looking into buying one, but unfortunately we don't have a donkey walking around that grabs money, so we still have to see if we can find the funds for that, because they really don't come cheap. So, and then these are a couple of machines you already saw before. So after we've done the work on the PIXUL, we then move on, continuing in a 96 well setup, either to one of these robotic systems. So we have I think on ResolveX for more simple workflows, but then now OpenTrans OT2, and just recently we bought also the OpenTrans Flex, because that one has the ability to have the gripper and move your plate around in your robot. So really, again, increasing the throughput of the workflows. So now to the fun part. So using the PIXUL, I'm just giving some examples here. We also did some comparative study. So we compared different tissue homogenization methods. So we claimed euthanized mice, and we kind of took different types of tissue, either soft tissues like liver, lung, brain, then to a bit more muscly, tougher tissue like heart, but then even going further to like cartilage and bone, when we also took ears and the tail of the animal. So here you see how much tissue we collected. This is wet tissue, 5 to 10 milligrams of wet tissue, washed it a couple of times. And then we also moved on to the STRAP protocol. So we collected it in a 100 microliter of STRAP buffer, the composition you see here, and then did the appropriate amount of replicates and with different homogenization methods. So of course we have the ultrasonication using the PIXUL with the method that is highlighted here. Then we also used different bead beading methods or bead milling methods, using either tubes or plates and with different bead sizes, so 3.2 and 5 millimeter beads, but then also looking at dissociation methods, dispersing and downsing. But an important remark here is that with any of the other methods, we also included again a PIXUL step just before, just because of that DNA sharing. So what we basically did is compare PIXUL alone with these other methods followed by PIXUL, because if you use them as such, you have the problem of the snotty-like samples due to the DNA that is in there. So we then put it in our workflow. So we had it in the Estra buffer, heated it for a while, and then compared either PIXUL with the other lysis or lysis methods, and then run through the STRAP protocol, so reduction, alkylation, and then the digestion on column. We analyzed those samples with a fairly simple setup, just to not to lose much time. So it's just a 1 hour run, nothing special, but really to compare how much protein we get from these tissues. So what you see here again, mentioning that with the other methods, we still use PIXUL after that. So if you, for example, look here at these softer tissues like brain, heart, liver, and lung, the PIXUL actually performs very well on its own. So either if you do, for example, bead milling or downsing or dispersing, I mean, there's not always a real added value of using that followed by PIXUL. So what we see is that you can actually go directly on the PIXUL and then move on to STRAP protocol. Of course, that doesn't go for ears and tails because these tissues are simply, yeah, they're too hard. So there we use the other methods, but keep in mind again that these are also followed by the PIXUL again. So then for these, it looks like an actual crushing of the tissue with beads is still necessary. And then I want to dig a bit deeper in one case study we have here, which is on amyloidosis. So the core facility is now part of an initiative called BAMICON, and this is funded through VIP grant challenges. So they have these special funding programs for really challenging projects. For example, amyloidosis, which is a fairly rare disease. So what we really aim to do is to better understand the disease and also improve the typing of the disease through proteomics. So what is amyloidosis? Well, it's an accumulation of aggregated proteins in different tissues and organs, which eventually leads to failing of the organs. And it's quite a severe disease because the prognosis is really not that good. I mean, from diagnosis, you usually have a couple of months to years after the diagnosis. So it really needs a good typing in order to have a good treatment and to extend the life expectancy with the disease. So you have different types. You have primary, secondary, hereditary, and wild type, depending on the protein that is aggregating in the tissue. Diagnosis is usually done through a biopsy, so a piece of tissue that they take, sometimes also in blood, urine, and now we are looking into implementing mass spectrometry, proteomics here as well. Treatment is sometimes in dealing with symptoms, and sometimes they can be treated with stem cell transplants, or sometimes the only real cure is like an organ transplant, or so not a very nice disease. So, for example, for an AL amyloidosis, you have antibody light chains that are accumulating, for example, in the heart muscle. So you have got like a B cell clone that are just producing a lot of these light chains, and these are then forming amyloids in between the tissue and hampering the function of that tissue, which leads to multiple symptoms. So what is done now? Well, usually they take a biopsy, they look under the microscope, they stain it with Congo red to confirm amyloidosis. But then for typing the amyloidosis, they rely on immunohistochemistry, but which is not always specific or sensitive enough to really have a good typing. So that is where the workflow really hampers, and where mass spectrometry proteomics can help. So this is our current workflow. So we have a PhD student, which is actually a pathologist working on that on the Lore. So she improved our workflow for these type of samples, let's say. So it all starts with laser capture microdissection of the amyloids themselves. So and that's, we also bought now a new laser capture microscope, the Leica LMD7, which we use for this, but also want to use later on, for example, for deep visual proteomics. That was also briefly mentioned by Alexander. So a couple of years ago, we dissected whole tissue, so micro-dissected tissues, which contain healthy tissue with the amyloid deposits. And this was already quite okay, but now with these new ultra-high sensitive machines and with our current workflow, we really cut out the amyloid deposits themselves, collect them until we have roughly 60,000 square micrometers. And then again, like you see here on the right, this is currently our workflow on the PIXUL. So we put it in 50 microliters of lysis buffer. We paraffinize overnight, so because these are all FFPE tissue, so not fresh tissue. Of course, this method also works for fresh tissue, but if you can do it on FFPE tissue, that's a big advantage, because you have a lot of these samples that are stored and you can do retrospective studies. So we put them in the PIXUL, 5 minutes, and then do a quick spin down, and then again another hour at 80 degrees. Then again on the PIXUL, then centrifuge, and then we continue to reduction, alkylation, and tryptic digestion. And then we run it on our timsTOF Ultra, now Ultra, in DIA. So we use DIA passive for these samples, for most shotgun samples actually, and which we then analyze with Diane and process the data through R with our own scripts. So this is, for example, an immunohistochemistry staining of an TTR in heart tissue, which was cut out. And then we do the workflow. Roughly, so depending on the tissue that we cut out, we get somewhere between, let's say, 250, depending on the size of the deposits, through 2,000 proteins. And just keep in mind that what we are cutting out is not actually tissue itself, not cells, but these are protein deposits which contain, let's say, a kind of a metaproteome that is attached to that. So what you see here, for example, so this is a real result of a tissue that was cut out, and you see all the way on top, you see the transthyretin popping up with a very high abundance, because that is indeed the culprit protein here. And that's what I mean with typing. So we eventually put this data in our scripts. And then we get these nice curves. So we rank them on abundance, on the IBAC value, and then we look at certain signature proteins that tell that we have indeed amyloidosis, and then really look at the culprit protein that tells you which type it is. And this method is really kicking off. It's really starting to work. Because it's not always that easy, because you're basically looking at aggregated proteins, and what you want is to dissolve those aggregated protein. So, and that's why the combination with the PIXUL and STRAP is very good here, because you have both things working together. You have a very good solubilizer SDS, but then also with the sonication, you help in dissociating and help dissolving those aggregated proteins again in solution, and then be able to process them. Because what you don't want is to spin them down again, of course, because then you lose them. So it's not just about typing. So in this case, we type by just cutting out the amyloid deposits, but we're now also really cutting out, for example, the periphery of those amyloid deposits, really having spatial information. So instead of going through the, well, you know most of the details of the method, but we published these as book chapters. So this was the first one that was published on the macro-dissected tissue samples, FFPE, using the PIXUL in combination with STRAP. But now very recently, this was also published, which is the tailored or the adapted workflow for micro-dissected FFPE tissues. So the example there is on amyloidosis, on amyloid samples, but we have used that same workflows for all kinds of micro-dissected samples. Either it's brain, but now also in muscle, and even also one's tendon. And it all works. I mean, it's a good workflow for doing that. Having said that, if you, of course, we here combine deposits from the same tissue until we have around 60,000 square micrometers, but we're also now developing these methods to go lower. So if you really go very low, I wouldn't say in tissue to single cell, but like a couple of cells or like tens to hundreds of cells, then, like I already mentioned in the beginning of my talk, we don't usually go to the PIXUL because we use much smaller containers for that. We hardly ever go to 96. We try to work in 384 plates. So we actually did the same exercise as Alexander mentioned. So we sorted out also cells. It was 500 cells in that case. In that particular test, we went to the MPOP protocol. So it's a minimal proteomic sample per protocol, just a one-pot reaction, because there really the challenge lies in not losing your sample through adsorption. So the DDM there helps, but you really want to minimize transfer of sample to because then you lose protein. Having said that, also the STRAP, so for that we use the micro STRAP. Also in a big kind of a disadvantage here is that Protify hasn't yet launched their plate setup for the micro STRAP, which is a pity because that would really help in having the plate based setup. So this was it for my part. Like I said, it was just a glimpse through an example. If there are any questions, please shoot.
Thank you very much, Simon. It was very interesting. There is one question. Can you talk about how you or collaborators get the cells from the culturing dish to the PIXUL 96 well plate? Do you lyse and transfer, or so it's a...
The tissue cuts, you mean?
No, I think it's culture cells.
Culture cells, so we often ask the customers to provide us with cell pellets. So either they provide it in vials most often. So what we do is add the STRAP buffer directly to the cell pellet, dissolve it, and then transfer it to the PIXUL plate.
So at the core facility, you receive cell pellets, in fact.
Most often, if it's really cell cultured, that is what we ask of our customers, to provide washed cell pellets. It can be in a plate, but it can also be in vials. Yeah, but that also always depends, and if it's, for example, tissue, it depends on the laser capture microscope they use, because some of them it's in vials that you capture the small laser captured tissue. Now the one that we just bought also enables collection of the tissue cuts in 96 well plates as well, so that's also an advantage there.
I have a question also regarding FFP samples. You said that it's better to use FFP, but I think you said that regarding the FFP collection, you can find
No,
the
it's not. Yeah,
hospital?
yeah. It's not better in a technical. It's just it's convenient that you can use the
Yeah.
FFPE. That's
Okay.
what I meant. Yeah, yeah, yeah. But actually,
But
we recently had a comparison with it was ovarian tissue. Where they did the comparison between fresh and FFPE, and the difference was actually very minute. There was almost no
okay.
difference. And then people, yeah, they decide to go for the FFPE, of course.
Mm. And do you know by experience if you lose some specific information? I don't know, for example, secondary modification of proteins or certain group of protein?
We usually don't. But we do take some modifications into account because you do get modifications on the lysines. It's methyl and methylylation. But we do take them into account in our data analysis workflows, although we actually also don't often see them. And there also we expected to see a higher number of missed cleavages because of the lysines that are modified, but also there it seems not to be a real problem, and the deparaffinization step also works quite well. And I think it has something to do also with the combination with using the SDS buffer, the heating step, of course, and also the PIXUL sonication.
And regarding the aggregates in the amyloidosis samples, you were able to like break this aggregate to then process your samples for mass spec.
Yeah. Yes.
Okay.
So, well, just keep in mind, so you also have other types of amyloid deposits, for example, in Alzheimer, and these are more challenging to work with. These are really, it's like they always say, it's an insoluble mess. Then it might you might consider having extra steps, for example, an acid hydrolysis step to really destroy the amyloid deposits. In this case, we also compare different workflows, but it appears that our current workflow is still the best. And if we don't have to do these extra solubilization steps, yeah, all the better for us. But just keep in mind that you have different types of amyloid deposits. Here it's systemic amyloidosis, and this method seems to work very well for that. If you go, for example, for brain amyloid deposits, for example, in Alzheimer, it might be good to have some more harsh solubilization steps there. Yeah.
Okay. There is two other questions. So just to follow the previous question in the chat, do you have issue with genomic DNA when transferring your cells from tube to the plate?
Normally not anymore after PIXUL.
Okay. And last question, when using PIXUL, would you recommend using the adhesive cover or silicone sealing mat?
I didn't understand, sorry.
Would you recommend using the adhesive cover? So the sealing, like the adhesive that you use with the plate, or rather use a silicone sealing mat?
I think we use the silicone sealing mat. Yeah.
Ah, okay. So now you're using the silicone
I
sealing
think
mat.
so. Yeah, I should check. I mean, but I'm not 100% sure, but I should check.
So you don't have any like drops sticking on the sealer. Everything stay in the well.
But we spin down here after we do the sonication.
Yeah, it's also because during a demo, what we saw is that when there was pieces of tissue, not for cells, but really for tissue,
Oui.
during sonication, you have some tissue pieces
Right. Yeah.
stuck in the sealer, and they are not lysed because they stay on the sealer.
No, to my recollection, we don't have that, no. But it's also, I think the volume there is also important, not to go too low in volume. Well, for these amyloid deposits, we haven't seen it before because we always have consistent results between the samples. There is variation, but the variation is usually due to the type of tissue or the type of amyloid, and not necessarily because of we lose it. Because in that case, using a laser capture microdissected tissue, you wouldn't see it actually if it's if it got stuck on the cover.
Yeah. Okay.
Yeah.
Another question, how much trypsin are you using for 60,000 micrometer square tissue? Does PIXUL help to minimize miscleavages?
That's a good question. I don't know, because we always use it for these FFPE tissue. So I cannot say not using it. So regarding trypsin, it's... Yeah, sorry.
So in fact, when we tested the PIXUL in the VIB, they compare the probe to the PIXUL, and the control they did was also the percentage of miscleavage, and it was similar with the probe and with the PIXUL. I can't say with other kind of sonicators, but it was comparable.
Yeah, well, we don't have issues with it. That's the only thing I can say with miscleavages, because that's something we always track in every sample. We have an automated QC system that gives these parameters of every sample we measure, and we compare it to a QC sample. So miscleavages is, even in these FFPE tissue samples, it's not an issue. And either with PIXUL or, but like I said, we don't often use classic sonication anymore. Actually, it's kind of replaced it. The only thing that we still use is bead beading, and then that's really if we have really challenging samples. But we actually also still use it for yeast and bacteria. That's why I also liked the things that Alexander showed. If we can do it using PIXUL with different buffers, that would be very helpful already for us. It's not the biggest chunk of samples we have in our facility, but still we have them, and it would again streamline our processes even more.
Okay. Is there any more questions? Last chance for you.
The next thing that we will try actually is plant samples.
Yeah.
That is something that is on our agenda for next, because we're also looking in spatial proteomics in plants, so laser capture microdissected plants tissue. That is actually next on our to-do list to see what the PIXUL can do there in small amounts of plant tissue, like root or leaf. Just to mention.
So I think what will be difficult is usually you have like a peptidic wall around the cells.
Yeah.
But I mean, with yeast, you also have this kind of issue. So maybe you can try a protocol close to the one you use for yeast.
Mm-hmm. Yeah.
Okay, so thank you very much, everybody, for joining us. Thank you for your questions. Thank you, Simon and Alexander, for your very good presentation. I was really happy to listen to you. Have a good evening. This presentation was recorded, so you will all have access to listen to it again if you want. Have a nice evening, everybody. Thank you very much.
Thank you. Bye-bye.